Hello, this is Jungmin Lim from the IBS Center for Molecular Spectroscopy and Dynamics and Korea University. Today, I'd like to introduce our recent research on bioimaging microscope using the Mirage effect. This photo is a hovering ship reported on the BBC and was taken near the corner. Due to the temperature change of the air between the boat and the photographer, it looks like the boat is floating in the air. Yeah, this is the Mirage effect. Based on this, we developed a novel bioimaging method generating the Mirage effect on the top of the slide glass. Now, let's talk about the actual experimental method. As a property of the material in the past do not change, the laser light keep its original directions. However, as shown in the figure here, if the temperature is changed by the IR absorptions, the light path is deflected. Then we could catch that by the photodiode detectors. If we could control the IR light precisely, distinctive temperature change near specific chemical structure is feasible even in the living organism. And the intensity of the deflected beam is proportional to the absorption cross-section and the population of the target chemicals. It has some advantages. Since we measure the deflected beam and block the original beam path, it's relatively free from the random scattering, somehow reduce the background levels. It also free from the photo damage and easily avoid the water absorptions. And it has sub-nanometer spatial resolution with the visible probe, and in the same time, it has the chemical selectivity with the mid-IR pump. After all, it is an excellent measurement tool with the high chemical selectivity as well as the high spatial resolution. Let me move on to the experimental data here. Visible probe light shown in blue deflected and reached to the detector when the mid-IR photon is observed by the sample and the temperature around is changed. As shown on the right-hand side, it can be seen that the measurement of polystyrene bead with a diameter of 1 micron is possible, and especially it showed that the separation or distinction of two closely located beads is also feasible. In addition, it is also possible to measure the nanostructures as small as 500 nanometers with the highest signal-to-noise ratios in here. In our knowledge, this is one of the most advanced technique in terms of spatial resolution for IR microscope in far-field regions. And then we try to apply this technique in the observation of the biosamples, as shown in the FTIR spectrum for human breast carcinoma. There are a variety of chemical structures and dynamics to be observed. As a test sample, we prepared neural cells, including neuron and glia. Here we also expect to observe the viscous transport through the axons. This is a bright field image of the oligodendrocyte, one of the glia in neural cells. To make the bridge among the neuron and axons, they have such a long arms and branches. In our photosomal measurement, we measure the photosomal contrast with the 1650 wave numbers, excitation for protein amide band, showing signals are broadly distributed at the central regions of each cell. We also see that the strong signals might be originated from the nucleolus. Moreover, the weak phototoxicity of this method allows a quite long time measurement, more than a day. So as you can see in here, we can clearly observe the cell division process. The photosomal contrast are also localized and delocalized throughout the division processes, we presumably thought that this is indicating the protein localization behaviors to support the DNA replication and division processes. In the case of neuron, the vesicle transport also through the axon branch are also observed. Like here, from the starting point to the middle and final point, the protein-rich vesicles moved. In terms of numbers, they moved about 10 to 20 nanometers per second with some kind of directional movement. So far, we observed various cellular behaviors with our mid-IR photosol microscope. As you can see here, the cell can migrate and they pass away naturally. So this is all. If you have any comments, question, or anything, please contact us with the email here. Thank you.